Hello, I'm Dr. Rikedu with the Department of Anatomy and Cell Biology of the School of Medical Sciences, University of Cape Coast. I want us to look at the male and female reproductive embryology, but this is going to be more female biased because we've covered that of the male already. So we just want to compare what the female reproductive embryology will also consist of. So the outline will be, we are looking at the gonads, but this time around only the ovaries, since we've covered the testes already. We're looking at genital ducts, in this case the uterus, vagina, and oviduct, because we looked at the seminiferous tubule, the straight tubule, the red testis, the efferent ducts, the epididymis, and then the vas deferens in the male. Then we look at the external genitalia here, labia, majora, and minora, mons pubis, and then clitoris, because we've looked at the labioscrotal swelling that forms the scrotum in males. We've looked at the retral folds, which will close, and you also have the phallus, which becomes the glans penis, and then you also have the penis. So the gross anatomy of the female reproductive system will look like that. It's just like somebody who has stretched the upper limbs, wide apart. Okay, so you have the fimbri, infundibulum, you have the ampulla, then you have the rest of the fallopian tube where you have the isthmus and the intramural portion as well. So that's the uterine tube. And that's the ovarian ligament, and that's the ovary, which is a gonad. So two of these will become ovaries. All right, and that's the uterus. You see what forms the uterus. That is the cervix. Then you also have the external os, obviously, but we are looking at the vaginal canal. There we look at all the embryogenesis of these parts. Very, very interesting. So let's touch on the embryogenesis of the ovary. So as we did for the male, we know that from the epiblast, we are going to have primordial germ cells migrating through the yolk sac and then going through the hindgut all the way to the gonadal ridge or genital ridge, where they will get incorporated into the primitive sex cords. So again, we look at that migration. The arrow is telling you the direction where these blue spherical masses, which are actually primordial germ cells, are going, and they are actually destined to come here as the epithelium of the gonadal ridge, which has started its extension into the mesenchyme and therefore the medulla to form the primitive sex cords. What is important to note is that in the male, this will remain, the primitive sex cord will remain, only the cortical portions will degenerate, but the medullary ones will be maintained and differentiated, okay, into seminiferous tubules with these primordial germ cells embedded in them. So that is what is going to, the primordial germ cells are the ones that are going to start forming the spermatogonia. So we looked at spermatogonia A, the dark one, and then the light or the pale ones, and on. But in the female, the primitive sex cords are going to degenerate, and then a secondary sex cord will emanate from the cortical region and then continue its differentiation. So note that difference. So this is what we are talking about at this point, where you have the gonadal ridge, the epithelium of it extending inward, and you can see primordial germ cells. Some of them have been incorporated into that primitive sex cords already, and then in the males it will canalize and then continue its differentiation at the medullary region, while the cortical regions are going to degenerate. In the female, you are going to have secondary sex cords. These are the remnants of the primitive sex cords, and they are degenerating. So that is what it means, that in the cortical region, you are going to have secondary sex cords developing in the female. Okay, so that's a point to note. The rudimentary tunica abugenia is also there. So the absence of the Y chromosome and the presence of the X chromosome contributes to femaleness. The ovary has two main parts, cortex and medulla. The ovary is not identifiable histologically until approximately the 10th week, prior to which we call that the indifferent gonad. So a different or indifferent gonad. Primitive sex cords proliferate and penetrate deep into the mesenchyme to form medullary cords. Indifferent gonad is where the primitive sex cords, also known as gonadal cords, are still connected to the epithelium of the gonadal ridge with the cortex and the medulla. Primitive sex cords disappear and are replaced by vascular tissue. Okay, the remnant in the medulla will form rete ovary. Then you have secondary cortical cords form, increase in size, and primordial germ cells are incorporated in them. These cords break up later into clusters to form primordial follicles containing oogonium, which are actually coming from the primordial germ cells. So note this. So the primordial follicles are as a result of the breakup of the secondary cortical cords. And within them, the primordial germ cells would have incorporated themselves into that secondary cortical cords. So once they are broken apart, they are going to become spherical in nature. And within them, you have the oogonium, which is actually a cell of the primordial germ. The primordial germ cells are the ones that have now changed their name to oogonium, okay, within the primary follicle. So the follicles will be surrounding the oogonium. So this is what you are going to see. So that is a follicle with the primary oocyte inside, which means it has even developed further. All right. But remember that this oogonium would be in utero. Before the female is born, you have the oogonium there already. And then they start their meiotic division and are arrested at prophase. Okay. So by the time the female is born, you actually have primary oocytes, okay, there. So primordial follicle will contain primary oocytes, and then primary follicle will also contain primary oocytes. Secondary or antral follicle will contain primary oocytes as well. Then tertiary or mature Graafian follicle, just prior to ovulation, are going to contain the secondary oocyte. But by that time, it would have finished its first meiotic division, okay, and started second meiotic division. So once it finishes its first meiotic division, then it changes from primary oocyte to secondary oocyte. Then after fertilization, you have mature oocyte, because at that point it would have completed its second meiotic division. Okay, remember that at the point where a secondary oocyte has started second meiotic division, but it is also arrested at metaphase. So this is how the whole process occurs. You have the cortical cords there. That is the surface epithelium. That is the mesonephric duct, which is more pronounced in male, and that's the paramesonephric duct, which we see more pronounced in female. So you can also tell that the medullary cords of the primitive sex cords are degenerating, okay, while the cortical ones, which are the secondary cords, are differentiated and they've been incorporated into by what? Primordial germ cells, which becomes the oogonium. Then later on, primary oocyte. So now you have primary oocyte there. Those are follicular cells. And the follicular cells are the secondary cortical cords, which broke into clusters. All right. So thank you, and I hope this video helped. This is just part one. We'll be looking at part two for the genital ducts in female, and then eventually the external genitalia. Thank you very much. So we want to now look at the female genital ducts. The paramesonephric duct, also known as the Mullerian duct, is very important here. So the paramesonephric ducts develop lateral to the gonads and also lateral to the mesonephric duct. So this is the very beautiful colored paramesonephric duct, which are lying lateral to the mesonephric duct and also lateral to the gonads. Okay, so that is it. It always reminds me of the stethoscope. Okay, so the wings of the stethoscope, and then where you have the knob, also very indicative of that. Paramesonephric duct fuses in the median plane to form a Y-shaped uterovagina primordium, where you find a uterine canal. The unfused cranial parts form the oviduct. So that is what you have here. The unfused portion here will form the fallopian tubes or oviduct, and the fused portion will form a Y-shaped structure. Okay, that will become the uterovaginal primordium, which means that it's going to form the uterus and a portion of the vagina. That's why we call that uterovaginal primordium. And you can tell that this actually looks like a stethoscope, where these two portions are the part that will go into the ear. Okay, and then you have the sensor where you use to check the heartbeat and all the pulse and all that. So always remember stethoscope when you see you see paramesonephric duct which has fused. So again, you have the paramesonephric ducts which are fusing in the midline to form the uterovaginal primordium, within which you have the uterovaginal canal, and then this is the urogenital sinus. You remember this in the renal lecture. Okay, so urogenital sinus, portion of the hindgut and the cloaca region. The uterovaginal primordium projects into the dorsal wall of the urogenital sinus, and once that happens, it is going to produce an elevation called the sinus tubercle or Mullerian tubercle. So we are saying that this uterovaginal primordium has contact with the urogenital sinus, and where the contact is made is going to produce a projection known as a sinus tubercle or Mullerian tubercle. So the sinus tubercle will form the sinovaginal bulbs, which will later become the vaginal plate. So remember, these are the paramesonephric that they fuse to form a Y-shaped structure, and then this is the urogenital sinus, and then the contact that the Y-shaped structure of the paramesonephric that has with the urogenital sinus will form a tubercle known as the sinus tubercle. And it is this sinus tubercle that will form vaginal plate. So that is it, sinovaginal bulbs and then a vaginal plate. Eventually, the vaginal plate is going to canalize to form a lumen. Okay, that will form the vaginal canal. So here again, we have sinovaginal bulbs where the sinus tubercle was occurring. So that is what happens. Then you have the fusion of the paramesonephric that will cause this peritoneal fold to form broad ligaments. What are some of the things that may go wrong? You can have uterus diadephis with double vagina, which means that the paramesonephric that joined did not, join well, and then there was no degeneration of the septum that borders both, tubes. So this is supposed to degenerate, and here you can see that the formation of the uterus, that's the joining of the two paramesonephric, that was not too complete. Here, that is not bad, it's okay, but there's an indentation here. That's a projection towards the lumen. But you have a very nice vaginal canal form because the septation in the middle has degenerated. So you have just one vaginal canal. Here you have two, okay, and then more or less two portions of the uterus. Then here again, the joining was not too well done. However, you have one lumen there. So that is the uterus bicornis. The previous one is uterus aquatus. And then the first one is uterus diadelphis with double vagina. You can also have uterus bicornis unicolis with a one rudimentary horn. So that's the rudimentary horn, which is the other portion of the paramesonephric that was supposed to have joined. Okay, it did not do its migration well. Only one was able to complete the migration, and so you have just a slender uterus here with the vagina canal, and then here you have the atresia of the cervix. So there's complete blockage. Okay, even though the uterus is well formed, and you also have the vagina, but there's no continuity, no communication. You also have the open part of the vagina here, and then atresia of the vagina here. So there's no vaginal canal that goes into the portion of the vagina. So you have to note that the formation of the vagina is coming from two sources. The upper portion will be from the paramesonephric duct, and then the lower portion will be from the urogenital sinus. So let's note that. So in summary, we are going to see that paramesonephric duct plus paramesonephric duct will give us vitreovaginal primordium, within which you have the vaginal canal through to the uterine canal. Then you have the vitreovaginal primordium contacting, for want of a better word, or impinging on the wall of the urogenital sinus to form the sinus tubercle. It's also known as the Mullerian tubercle. Then this is going to form a sinovaginal bulb, which will later form the lower portion of the vagina. Remember, the upper portion of the vagina will come from the paramesonephric duct. The lower portion is coming from the urogenital sinus. Thank you very much. I hope this video helps. When it comes to the development of the external genitalia, we are going to basically look at what the vagina will look like and what is going to form it. Early in the 4th week, proliferating mesenchyme produces a genital tubercle in both sexes at the cranial end of the cloaca membrane. So the cloaca membrane, you are looking at the portion of the hindgut where you have the cloaca, right? And then you also have the membrane covering the cloaca, which is the cloaca membrane. You have labioscrotal swellings and urogenital folds which develop on each side of the cloaca membrane. The genital tubercle will elongate to form a primordial phallus. So this is what we are talking about. That's the genital tubercle, okay, forming the primordial phallus. That's the genital swelling or labioscrotal swelling. That's the cloaca fold, and that's the cloaca membrane. The cloaca fold now becomes the urethral fold, also known as urogenital fold. So that is the phallus, okay, the projection of it in an indifferent gonad. So at the 7th week, there's indifferent gonad. You can tell that the gonad or the external genitalia will be male or female. You have genital tubercle, that is the genital fold, that's the genital swelling, that's the urogenital sinus, within which you will have the cloaca membrane. So by the 9th week, things are changing gradually, but it's still not complete to determine whether it's male or female. So you have the phallus there, genital fold, urogenital sinus, genital swelling, which is also called the labioscrotal swelling. So we are interested in this pathway for the female. We've covered that of the male already. So the female, by the 12th week, clitoris forms from the phallus. So it has now become smaller. Then you have a urethral orifice within the cloaca membrane. You have the genital sinus. Then you also have the vaginal orifice. Then lateral to that, you have the labia minora, which is actually coming from the genital fold. Okay. Then you have the genital swelling forming the labia majora. Genital swelling. Yeah, this one, also known as labioscrotal swelling, will form labia majora. So again, you have the clitoris, labia majora, labia minora. Then you have the vaginal orifice and urethral orifice, and then the anus. So indifferent gonad at 7th week. Number 1 is the developing penis or clitoris because it is indifferent at this point. Number 2 is urethral groove. Number 3 is urethral fold. Number 4 is the labioscrotal swelling, which will later become the labia majora in the female, but scrotum in the male. By the 10th week, the female fetus, we have the number 1 being the clitoris, external urethral orifice is number 2, where you have the cloacal folds joining. Then it's going to completely cover that in the male, but in the female, you still have that orifice opening, okay, opening into the urogenital sinus, urethral fold, which will become the labia minorus or minus, number 4. Okay, remember that in the male, that one, if it doesn't completely fuse, you're going to have hypospadias, okay, urethral fold. Then you have the labioscrotal swellings, which become the labia majora, number 5. So in this picture, in utero photograph of the genitalia of a male fetus at 12 weeks, note that the urethral folds are fusing and that the scrotal swellings are enlarging to merge in the midline. So this is what is occurring. These ones are enlarging and they'll fuse in the midline. So the genitalia of a female fetus at 11 weeks, note that the urethral folds, which will become the labia minora, have not fused yet. So these ones have not yet fused, unlike here that's almost fused completely. They have not fused, and that genital swelling that are formed the labia majora are widely separated. So you can see this one is going to form the labia majora. They are widely separated compared to that of the male, which are trying to close up and join each other, okay, to form the scrotum. So the inner urogenital membranes will later rupture, forming the anus and urogenital orifice respectively. In the female fetus, the urethra and vagina open into a common cavity known as the vestibule. So you can have some problems. This is the normal thing that should go on: urethra, bladder. Remember that the bladder is forming from a portion of the urogenital sinus. And because the urogenital sinus also is connected to the reproductive system in a way where you form vagina and all that, sometimes it can be connected as you have here. So you have persistent urogenital sinus, that's the vaginal canal opening into that of the bladder. That is not too good. So in this normal one, you have the uterus, vagina, and the rectum. The genesis of external genitalia, there's nothing there. So you may not have external genitalia at all. No phallus, no clitoris, no penis, no labia majora, minora, scrotum, and the like. So in summary, we are saying that primordial germ cells will migrate through the yolk sac and then come all the way through the hind-gut towards the gonadal ridge. When they get there, they incorporate themselves into the primitive sex cords that are emanating from the surface epithelium. In the male, this continues. The primitive sex cords grow towards the medulla, and then the cortical portion, which are connected to the surface epithelium, will degenerate, and only the medullary ones will be maintained with the primordial germ cells incorporated into them. Okay, but in the female, they will all degenerate. Whatever will be left of the medullary portion is the reti ovary. As for the cortical portion, everything goes away and it is replaced by a secondary cortical cord, which will be incorporated with the primordial germ cells, or the primordial germ cells will incorporate them, okay, and then continue the development as follicles with oogonium. So the primordial germ cells become oogonium, and then the follicles will be the clustering of the secondary sex cords. Then we also said that the paramesonephric duct will join medially in the midline to form a Y-shaped structure, and then you end up having a portion, the uterovaginal primordium, which will now become a portion of the uterus and then the vagina. Then at a point where the paramesonephric duct and therefore the uterovaginal canal have contact with the urogenital sinus, there's going to be a sinus tubercle, which will become sinovaginal bulb and then a plate. And the plate will now canalize to form the lower portion of the vagina and the vaginal canal, which will go through that of the upper portion of the vagina into the uterus. All right. We looked at the malformation of the uterus and the vagina. Thank you very much. I hope this video helps.